Well, we're very happy to have Lars Bejder here with us for a bit of an extended talk. Lars is the director of the Marine Mammal Research Program at HIMB. He has a PhD from Dalhousie. Before joining UH, he was based at Murdoch University in Western Australia for 12 years. He has studied cetacean biology, ecology, and conservation in New Zealand, Australia, Hawaii, Florida, California. Has over 100 research journal articles on cetacean behavioral ecology, quantitative methods, and impacts of human activity. On the international stage, Lars serves on the International Union of Conservation of Nature's Cetacean Specialist Group and is a member of the Scientific Committee of the International Whaling Commission. His talk is entitled "The Use of Innovative Technologies to Study Resident and Migratory Cetaceans."
Good morning. Thanks all, and thanks for the kind invite to come here today and speak. Really nice to be here. I have somewhat recently joined UH. I've been here just shy of 2 years, and now out at the Hawaii Institute of Marine Biology. There is a long history of marine mammal research at HIMB, historically focusing on whale and dolphin hearing and sound production. After my arrival, we've changed focus somewhat, and what I'm going to be presenting today is what we've been starting to work on in the last couple of years, particularly with the focus on new technologies, giving the theme of this symposium today. A lot of the work that I'm going to be presenting and talking about is of my graduate students, so thanks to them and not myself. So with the focus of technology in the symposium today, I'm going to be talking about two particular pieces of technology that we are using quite heavily. One is the UAS, so unmanned aerial systems or drones, and how we're using them to assess individual and population level assessments of free-ranging cetaceans. I'm also going to be talking about how we're using non-invasive tags, so suction cup tags, to study. Right now, we're studying the feeding ecology of baleen whales with some collaborators. So jump around from various projects to hopefully give you a bit of an overview of how we're using these technologies. The first piece that I want to talk a little bit about is humpback whales that are migrating between our Hawaiian waters here and up to Alaska. Particularly, we're trying to develop a bioenergetic model of what's the cost of migration and what is the cost of being a baleen whale. These are capital breeders, means that they subsidize their reproduction based on stored energy, which they acquire in Alaska during their feeding grounds. And the overall, focus of this research is trying to explore how climate change might change the dynamics of humpback whale populations. We are carrying out these studies with other colleagues around the world with different stocks of humpback whales and trying to compare these stocks between them, particularly by quantifying changes in body condition. They are moving a lot of mass throughout these big migration periods. A quick 101, humpback whales migrate between Alaska and Hawaii. There's about 10,000 humpback whales that come here during the breeding season throughout the island chains, and about 3,000 humpback whales are found in Southeast Alaska. That's a 6,000-mile return trip. Generally, when they leave Alaska, these animals they opportunistically feed very rarely during their migration, but very little, which means that they need to acquire all their energy while they're up in Alaska until they come back again 4 to 5 months later on. And that's a long time to go without feeding, particularly if you're a female that's giving birth to a calf and has to lactate, bring that calf up to a given size, and that calf have to migrate back up on its own. Now, I mentioned that we're using drones to look at body condition, and body condition is a really, really good indicator of health. Sometimes I feel like that polar bear, and Saturday mornings after a few drinks, I feel like that top runner up there. So body condition is a really, really strong indicator of survival and reproductive success, and really has strong connotations and input for conservation status of individuals and also for populations. Now, fortunately, with the proliferation of using UAS and also better lenses and better altimeters, we are becoming really, really good at quantifying body condition and body size. A lot of this work was pioneered by a previous graduate student of mine. I just want to acknowledge Frederick Christensen as this work. But generally, if you have a UAS equipped with a flat lens, so with no distortion, and we can attach very precise LiDARs, you're very good at estimating your height, and with a flat lens and you know your pixels within a picture, you can change or you can calculate absolutely length by knowing your pixel sizes. And by doing that, you can actually come up with some calculations of how big some of these animals are. Now, there are some assumptions, obviously, when we do this. We assume that these animals are conical shapes throughout their whole, or round shapes throughout their whole body mass, and obviously there are some imprecisions with that. But generally, we go from a two-dimensional image to a three-dimensional image based on these cylindrical, or these slices, if you will, and then we calculate the volume of each slice to come up with an estimate of the full volume of these animals. So we just commenced this last year. Our collaborators, really important to note here, is the Alaska Whale Foundation, also the Pacific Whale Foundation here in Hawaii, and researchers at Hilo as well. Martin van Aswegen is my PhD student who's really doing the grunt work here, so just want to acknowledge Martin. So over the last year or so, we've measured about 500 animals up in Alaska during the feeding period, and the last season we measured about 350 whales here off Maui. And these were preliminary seasons, and we'll be doing this over the next 4 years or so. So I don't work with hundreds and thousands of samples, but many of you guys do. But in my world, this is a pretty good sample size to start with. So let's just talk about it. What's the schematic, I guess, hypothesis of how a body condition of a Bolivian whale changes over a period of migration? So let's assume, so this is what we're calling relative scales. Now, poor body condition to a good body condition. And here we have the Alaskan feeding grounds and the Hawaiian breeding grounds. Generally, conceptually, humpback whales come back to Southeast Alaska in early April, and we assume, and we don't know, this is probably not linear, but again, just for the sake of argument here, that they certainly increase in mass throughout the 5-6 months that they're up there. They start migrating down, and for the ones that come to Hawaii, they don't all come to Hawaii. The ones that come to Hawaii presumably will lose condition while they're here, particularly the lactating females that, again, have to raise their offspring. And then by March, April, they start turning around again and go back up to Alaska. Now, just some preliminary numbers here from Martin's work. So here we have animals that we measured in Alaska in November. So if you just refer back to our schematic diagram, so that would be at the very end of when they're about to leave. So that's probably when they are as big as they're going to get. These estimates right now are, we've lumped males and females. We have the individual identities and we know the sexes of these animals, so we're actually going to partition the analyses based on males and females. But just for today, Martin has had many late nights just to get some preliminary data up, so thanks, Martin. So we're assuming here that on average when they leave, they're about 22 cubic meters. These are only adults. And when they get back to Alaska, in early April, they're down to about, what are we at? About 14 cubic meters. So on average, an adult humpback whale, from the cost of migration, meaning that when they leave here, go to Hawaii, and come back again, they've lost about 30 to 35% of their body mass. Again, this is averaging between adult males and adult females. When you look at within Hawaii, so we did January and March off Maui, you can see the kind of differences there are between just on their breeding ground themselves and how much they're losing here. One interesting part here, Martin was mentioning that some of the, he took a random sample here, but there were some other animals that he's subsequently done that we think are adult or pregnant females that are almost 30 cubic meters of volume. So some quite large differences, but clearly differences between the males and the females. And if you look at the scale here, again, this is just based on their time in Hawaii, and you can see how they're starting to lose condition. Now, we also looked at calves that were born last year off Maui between January and March, and then we found, these are not the same individuals, but the calves that were found in Southeast Alaska, and you have about a 350% increase in body mass over a 7-month period. So you can see how much energy, particularly for the moms, to fatten up these calves. Again, remembering that the moms aren't feeding as soon as they leave Hawaii or Alaska. Particular small little fun fact here, we were very fortunate in January, this is not depicted in this plot, we were informed of a very recent calf, as in within minutes, mom was still bleeding. We were able to go over and estimate that animal, and a very newborn within minutes is about 0.3 cubic meters. So showing that that's all the way down here kind of thing. So saying that these calves that we have there are probably a few weeks old already. Okay, so I guess point here is that this technology is allowing us to quantify changes in body mass quite readily, and something that we couldn't do 5-6 years ago. So it's really allowing us to get to some issues that we really never thought we were able to do unless we relied on whaling data, and we prefer not to do that. So we're quite encouraged with these initial results, and as sample size starts coming up and we're able to partition between age classes and sex classes, I think it's going to be quite interesting. And then starting sort of relating that to larger oceanographic features, it might shed some light on some interesting questions. Okay, so what I've shown so far is we're lumping across populations here, if you will. So we are going to be able to track individuals and how individuals change over time. And I just want to change focus here. On August 29th, these are numbers have just come out. This is in partnership with the Pacific Whale Foundation. On August 29th this year, 11 pygmy killer whales stranded off Maui. 4 or 5 of those were euthanized. 6 of them were refloated. Those animals are deep water species, but they stayed in very, very short coastal waters for about 3 weeks after that. Jens from the Pacific Whale Foundation used his UAS to go out there, fly same systems that we've calibrated, flew all those animals over a 3-week period. These animals should have been offshore foraging. Now, these are income breeders, so very different to capital breeders, right? So they don't. They feed and reproduce continuously, if you will, or feed continuously, and then are able to use those resources for reproduction, unlike the baleen whales that have this massive storage, meaning that these animals do not have as much storage capacity as baleen whales, which means that they probably need to feed continuously, right? Now, because we're able to look at the same technique, looking at the volumes of these individual animals, we were able to track how quickly the body condition of these animals changed. Christy West carried out postmortems of these individuals. These 2 here, I believe this one here and this one here, stranded the day after and they died. She carried out postmortems, and the stomach contents of those were very irregular. So really providing us some very interesting results on how fast these animals, income breeders, really lose condition. As another tool, using these tools for some quite interesting results. So we're encouraged by these kinds of opportunities. Okay, now I want to switch to, that's tracking individuals over time, if you will, right? Now I want to change focus, and this is a collaboration with JIMAR, which is a really good example of the JIMAR facilitating some of our work. Typically, marine mammal populations are of abundance are evaluated through either marker capture, which we heard about earlier on today, or line transect surveys. And typically, the power to detect changes in abundance estimates are very, very low for marine mammals, and they take a long, long time to do. They are quite labor intensive, quite expensive to do, and the trade and still the power to detect changes is quite low. Very often you need a very large change in abundance to actually to detect it. So there is a need to develop new approaches that might be able to give us insight into health of population. Now I'm not talking about individual body condition health, but health of populations in regards to demographics. And one of the things that we're trying to develop is a new technique to get an early warning signing of whether populations are doing well or not. And that's potentially looking at a population age structure. Now, the idea here is, and this goes for any, I guess, population, is that if you have a healthy population, you can assume some kind of distribution in the age structure, right? And these are some scenarios that PhD candidate Fabien Vivier, who's doing this work, has created. But the idea is that if you have an increasing population, you have many pre-reproductive individuals, and if you have a stable population, you have a different demographic age demography breakdown. This has been shown in the eastern tropical Pacific, where there was a large millions of spinner dolphins were taken as part of the tuna dry fishery. That data has been analyzed historically to look at how that population, the age class of that population, whether that's stable or what it would be compared to what is expected. Now, if you can come up with what the demographic background should look like if you know the age, the reproductive biology, and how often they have interbirth intervals. You can come up with a prediction of what that age class should be, or that breakdown should be. I think this was maybe about 1,000 individuals, and you can kind of get at here that age demography has certainly been changed through this bycatch situation that's happening with this population. So with that background, we're trying to come up with an approach to be able to go out using UAS to quantify the age structure of free-ranging cetaceans. Now, there's several steps in doing that, right? One, we have to calibrate our systems, which I'll speak to you about, and there's a poster on that by Fabien today. We have to calibrate our systems. How good are we at measuring the length of these animals? We also have to have age length curves, and with the idea is if you go out and measure the length of an individual, you're going to be potentially able to infer its age, and then the next steps is then you go into the demographic breakdown. Now, fortunately, from that ETP data, there are hundreds of thousands of teeth available from those spinner dolphins and length measurements. So we are recreating an age length curve for that population. It turns out that the morphology between that population and the ones we have here in Hawaii is about 6% difference. So I think there's some good take-home messages from that. But once you have those two pieces of information, we can then go out, estimate the length of individuals in the wild to come up with this age structure. But as the first step, as I mentioned, we have to calibrate our systems. And Fabien is working closely with the Science Center and JIMAR staff to do exactly that. He's working with Dolphin Quest and going out. They have 6 bottlenose dolphins in captivity, physically measuring these animals, every single morphometric measurement that you can think of, using 2 different types of UAS. So Inspire DJI 2, which is a Chinese product that NOAA is officially not cleared to use yet because Chinese are spying on us and we can't use them. And then the APH-22, which is the system that NOAA has been using. So we're not only just, so we're also comparing systems, but at the same time calibrating and seeing how precise we are. Fabien's going to be presenting some of these results, but a couple of the things that he's found already is that, so one of the things, so when an animal surfaces, typically you don't get the full body length, but you can get some proxies, and that one that he's using is the blowhole to dorsal fin length. He's also found that and also other work has shown that there's a direct correlation between that length and total length. There's an R-squared of about 0.92, meaning that if you can measure that length from using UAS, you can predict the length of the whole animal and so on. Now these are the differences. So these are 5 or 6 individuals that Fabian's measured, and I think the mean difference between the physical measurements and the UAS measurements is about 1 to 2%. So it's pretty good. And then when he's also measured the full length of the animals in captivity with the drones, and we're down to about 1% on average. So if you're trying to evaluate age or age class, I think this is a really good approach and seems pretty promising. So I think if this works, it's really going to help us move forward, not just with these populations that we have here, but in general, coming up with a new method to assess the health of these populations. So the next steps of that work is now to go off the Big Island. Through previous work, we know there's about 650 spinner dolphins off the Kona coast. We have a lot of information about their habitat use and so on and so on. So that's going to be our model species, which is the next step of that project. Fabian's work or poster is down there. So if you guys have time and have some more questions, he's the expert on this. So I'll pass you his way. Okay, so I'm going to jump over now to the last bit of the talk, which again using technology, but here we're both using UAS and suction cup tags to study the foraging ecology of Baleine whales. This work is in collaboration again with the Alaska Whale Foundation, UC Santa Cruz, and colleagues at Stanford. So the main thing here that we're trying to do, but let me step back. Humpback whales have many different ways of foraging. One quite unique way of foraging, and only certain individuals do this, is that they do this bubble net feeding that many of you probably have heard of, where they exhale in a round shape, if you will, and with the idea of trying to encapsulate their prey within that and then scoop the animals up within this tunnel that comes up through the water column. Now they prey on generally two different species, on herring, and typically that is a coordinated behavior, sometimes between 10 to 20 individuals, where we think individuals have particular roles in schooling up those fish. And then we have another technique where it's solitude animals working on zooplankton. We were able to tag 12 animals on a recent trip, 6 from the herring bubble netters, as we call them, and 6 on the zooplankton bubble netters. And what I'm going to be showing you today is the data that's partly been analyzed from the zooplankton bubble netters. So we're using a couple of different tags. The main one that we're using right now for this work is called the CAT tags. It's the Customized Animal Telemetry System tags. This tag has your 3 accelerometers, your pitch, your roll, your heading. It has depth sensors. You're able to look at fluke strokes. It has cameras, which is the really neat thing, is we can start corroborating our three accelerometers with actual what's happening, and hydrophones, so we can look at the sound production that these animals are actually producing while they're doing their capturing. We attach them with suction cups with a long pole. This is a little video of one of the animals on a patch of zooplankton. This is the first time we've been able to look at this stuff, certainly from the animal's perspective. We sped it up here a little bit so you can kind of see what's going on. And the drones from on top, where you can start this is 2 individuals next to each other working on a similar patch, as you can see. Now once you start looking at the actual sensors, the 3 accelerometer sensors in that, I'm just going to move forward to that, and this is where it starts getting interesting. So if you look at right here, it should be moving. Let's see. Yeah, so here you have the red line. That's what you're seeing in real time on the video. Right now it's on the pole, it's upside down. Once we get about another 10 seconds in, you'll see the tag being attached. The blue line is depth, and then you have speed here, and then you have what we call jerks. And down here you have the 3 accelerometers, which is the pitch, the roll, and the heading. So speed this forward a little bit. All right, so the tag goes on. Off you go, and you can start looking, corroborating what you're seeing on the tags with the accelerometers. Now, I'm going to fast forward it. So the blue line here is essentially a compass bearing going from minus 180 to plus 180, so meaning a 360-degree turn. So each one of these lines is one complete circle, if you will. And this tag stayed on for 18 hours. Really interesting is that this time of year, sun goes down about 10:20 p.m. The animals stop doing this at 5 minutes within that. The sun rises at 4:15. Within 5 minutes of that, it started the exact same thing. You presume that it's something to do with the vertical migration of the zooplankton at the time. Within that time period, it did more than 600 sessions of these loops. So you can start looking at foraging rates of these kinds of activities, linking it with the drone, the imagery with body condition. We can start looking at what is the rate of mass or body condition increase based on prey type, and then comparing that to the ones that prey on herring, which do not engage in this behavior that many times, repetitive behavior over time. And we can then start comparing that to the foraging ecology of animals that don't feed in any of these techniques, and then we can really start to look at what's best bang for buck as a foraging tactics, if you will. Now, I'm just going to move this. Oh, sorry. Move this forward. Try again. Clearly not a Mac person. So we move this here. So if you look, all right. So now you can see here, just before it starts turning, right? And just before it starts turning, it starts doing its exhale. You'll start seeing here now start pumping out. There we go, just before it starts turning. If you noticed the video you saw from top beforehand, there was a long line first and then you saw the circle, which means that that's exactly what we're seeing here. They start blowing and then they start engaging in their 360-degree turns. Now this is what's actually a schematic of what the animal is doing in that time. If you start looking here, this is quite interesting. The pitch is, it's heading up or downward, if you will, so it's a 2 or 3 degree up, so it's not actually going up in the water column very steep. Here you see the roll, so basically the tilt, so it's basically on a 45-degree angle as it's banking around, and heading, as you can see, keeps changing throughout each circle. When you get to the very end of it, you can actually see as it's coming up, you can see the zooplankton flying by the cameras as well. So really allowing us to answer some questions that we certainly haven't been able to do previously. And the last one I just want to show you, and this is some of the data that we're constructing, what that really looks like. Let's see here. Now there's a bit of drift. This is dead reckoning, meaning that there's no GPS on these tags. So these columns are actually completely vertical, but there's a drift in the way when we're doing these kinds of things. But it really gives a good depiction of how these animals are working these patches. The next phase of what we're going to be doing next year when we go up is we're going to be doing some prey mapping at the same time in these patches to see where the patches are and linking with that. Now, if you zoom out of this for 18 hours that you can really see, you can really clock that exactly with sunrise and sunset and see when they start and stop this behavior, which is really interesting. My daughter loves this cartoon. All right, so, So yeah, again, just getting back to technology that we're talking about. For us in the marine mammal world, we can certainly start getting into some things that we've never been able to do before. Not only very neat behavioral repertoires, which is really allowing us to answer some of these things, but more from a management perspective, starting looking at stock abundance and structure, which is one of the main mandates of NOAA, the Protected Resources Division. We can really help answer some of those questions and hopefully come up with some techniques that can provide some insight into how these populations are doing as an early warning. So, yeah, thank you very much. So the question was if there's a correlation between the precision of the altitude and your height of the UAS. Yeah. So if you're lower than 10 meters, your precision is less through the LiDAR system. It's a custom fit LiDAR that we put on. Once you go above 10 meters, and we've tested up to 120 meters because that's the level that we're allowed flying at, up to 120 meters, the precision is 2 centimeters height. Yeah, so it's a nice system. There are some issues that we're still dealing with. It's if you have your LiDAR system and there's a bit of wind, the drone's going to sit like this, meaning that your LiDAR system is going to point like that. So there's some calculations that both Martin and Fabian are really dealing with right now. But your precision of your exact height is quite good. Yeah.
Thanks again, Lars.